Hello students, today we are going to discuss about dihybrid cross. What is dihybrid cross? It is the cross in which individuals differing in two characters. That is, it is the inheritance of two pairs of opposite character or two pairs of contrasting traits. Here, what are the two characters? Seed shape and cotyledon color. Here in seed shape, they are either round or wrinkled. In cotyledon color, they are either yellow or green. And in pure breeding, the round and yellow is represented by capital R, capital R, capital Y, capital Y. Whereas in pure breeding, green wrinkled is represented by small r, small r, small y, small y. Here the round is denoted by capital R and it is the dominant character. Whereas the wrinkled gene is denoted by small r, it is the recessive character. Whereas in the cotyledon color, the yellow is denoted by capital Y and it is the dominant character, whereas the green color is denoted by small y, it is the recessive character. In dihybrid cross, Mendel chose these two characters, that is round yellow and green wrinkled. He crossed these two characters. In the F1 generation, he got all the offsprings are round yellow. And it will be explained in the Punnett square method. So here that round yellow is denoted by capital R, capital R, capital Y, capital Y. Here there are two gametes are found. That is capital R, capital Y, capital R, capital Y. Whereas the green wrinkled is denoted by small r, small r, small y, small y. Here also two gametes are found. That is small r, small y, small r, small y. Then we will explain by this Punnett square method. In Punnett square method, we want to draw two rows and two columns. Here capital R, capital Y, capital R, capital Y. Then in here small r, small y, small r, small y. When we cross this, all the offsprings are round yellow. That is, it have the genotypes of capital R and capital Y. So all are round yellow genes are obtained. He took two plants from the F1 generation. In the F2 generation, he observed the offspring contain four different characters. That is a round yellow, a round green, a wrinkled yellow, a wrinkled green. They occur in the ratio of 9:3:3:1. And this genotypic ratio can be explained in the Punnett square method. That is, the parental character round yellow, they contain the genotype of capital R, small r, capital Y and small y. It produces four types of gametes. Capital R, capital Y, capital R, small y. Then small r, capital Y, small r, small y. In the Punnett square, we using these four gametes in one side, that is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. And another parent also produce same four type of gamete. That is, it will be written in this side. That is capital R, capital Y, capital R, small y, small r, capital Y and small r, small y. When we cross these two characters, they produces capital R, capital R, capital Y, capital Y. Then when we cross these two, they produce capital R, capital Y, small y. When we cross this one to this, capital R, small r, capital Y, small y. Then it fuse with here capital R, small r, capital Y, small y. So in the first row, all yellow. Y means here capital R and capital Y is the dominant character, that is the round yellow. Then in the second row, when we fuse capital R and small y, capital R and capital Y, first cell we got capital R, capital Y and small y. Then second cell, when we fuse these two, we will get capital R, small y. Then in the third cell, when we fuse these two, capital R, capital Y, small y. Then in the last cell, we will get capital R, small y. Here, in the first two cell, we will, in the first cell only, we will get round yellow seed. Y means here the dominant character, that is capital R and capital Y is present. So, they are round yellow. And in the third row, we fuse these two. In the first cell, we will get capital R, capital Y, small y. Then second cell, when we fuse these two, capital R, capital Y, small y. Then third, when we fuse these small y, then small y. Then next to last row, when we fuse these two, small r, capital Y, small y. Then the last row, when we fuse these first two, capital R, capital Y, small y. Then when we fuse this one and this one, capital R, small y, small y. Then here, when we fuse this one and this one, that is small r, capital Y, small y. Then last row, that is small r, small y and small y. So when we observe this phenotype, that is here capital R and capital Y is present, then it is denoted as a round yellow. That is here first row fully, and here it also round yellow, here also round yellow, and here also round yellow. Then here also round yellow is present. When we count these, 9 round yellows are present in the, here also round yellow is present. So 9 round yellows are present. Then when we take the round green, that is it contains the capital R and green is denoted by small y. And here when we take the first row, here no round green seeds are present. In the second row, the second, that is round, that is capital R and small y is present. So it is a round green, and here capital R and small y is present. So it is also a round green in nature. Then here in the third also there is no round green. In the fourth row, here it is capital R and small y is present. So it also indicated by round yellow. So when we totally 3 round green are present. Next is the ringled yellow. It is represented by small r and capital Y. So in the third row, the third cell contains the small r, capital Y. The next cell also contains the small r, capital Y. Then in the fourth row, third cell contains the small r, capital Y. So totally 3 offerings is indicated as ringled yellow. The next is the ringled green. It is represented by small r and small y. It is present in the fourth row and the last cell. Only one offspring contains the ringled gene. Thus, Mendel's 9:3:3:1 ratio is explained by Punnett square method. Though two pair of characters combined in the F1 generation, but they become separate in the subsequent generation. Mendel dihybrid ratio lead to the formation of second law, that is the law of independent assortment. What is law of independent assortment? In inheritance of more than one pair of characters in a cross simultaneously, the factors which are responsible for each trait are distributed independently to the generation. In dihybrid ratio, the like this form. That is a capital R, small r, capital Y, small y. Thus, it forms the four gamete. That is, it follows the independent assortment. That is capital R, small r, capital Y, small y. It forms four gamete. That is the capital R, capital Y, capital R, small y. Then small r, capital Y, small y. Thus, gametes are present in the separate chromosome or randomly assortment taken place. So, four different types of gametes were formed in the subsequent generation. Next, we are going to see about why these seeds are occurring round or ringed. The molecular explanation is the round, that is the dominant gene is encoded by an enzyme called starch branching enzyme, that is SBEI. This enzyme is responsible for the conversion of unbranched starch amylase into the branched starch amylopectin, so that it occurs in the round pin. These are the dominant character, where in this gene level one of the mutation occurs, that is the insertion type of mutation occurs. Here the small or that is the recessive allele can be joined to the dominant allele, so that these are, that is the small or become recessive. Here it lost the function of the SBEI, that is the enzyme does not carry out the conversion of linear unbranched starch amylase into the branched starch amylopectin, so it occurs in the ringed shape, and it also absorbs more water. When it matures, it lost the water and it becomes ringed. Whereas in the dominant gene, that is the conversion of linear unbranched starch amylase into branched starch amylopectin contains some amount of water. It does not lose the water, so it becomes round in shape. So thus today we have seen about the dihybrid ratio, law of independent assortment. Thank you, students.